Okay, so again, just to review, I took the genes, the about 1,200 genes that were different between blood cells isolated from sickle cell individuals compared to normal. Um, from that, and this was compiled based on it being differentially expressed in at least 4 out of 7 studies that looked at that condition. Once I had these genes, and again, I used the median gene expression, I imported that list into IPA. This is Ingenuity Pathway Analysis. I don't know, I'm sure you're familiar with this, but I really like this data set because it is probably the deepest and best curated as far as relationships go between genes. So I really like it for that fact. That if we want to explore a gene in more detail, I can ask this program what is it known to interact with and see if a particular, say, upstream regulator or master regulator is affecting a lot of its known genes in my system. It's very good for that. This is the summary page, so I've uploaded it. It's basically showing me those things that are overrepresented in our gene list. Um, we can go to disease and functions, and I can size this by the number of genes. So the bigger the box, the more genes associated with that particular function. If it's blue, that means based on the gene expression, we would expect that pathway to or function to be inhibited. If it's orange, it's expected to be more activated. And you can see there's a lot of stuff down in genes that change in sickle cell compared to normal. A lot of downregulation of T lymphocytes, but I wanted to show you something here. We have in this box here, the fairly large box, out of the genes that we put in, we have 213 that are known to be associated with viral infection. And I can click on this box to bring up those lists. Not only that, but based on the direction of these genes and what is known about them, this will make a call saying that when this gene goes up in a system, it usually decreases viral infection. Same way with this. When this gene goes down, it's known to decrease viral infection. And you can look up any of those references to basically here are all the references that give you that information. There's much more evidence that this process is decreasing than increasing, which I think is very interesting. I don't know if this is due to based on differences in cell types, but um, I don't know. It's a very interesting phenomenon here, and that's kind of why I wanted to pursue this a little bit. We can get all of these genes, and I can add this to my list. We could make this viral genes, and I can give you this. Okay. Oh, there it is. Viral genes. Maybe you can look at this. We can actually make a subset using these genes, and I can actually analyze just this. But again, I think this is kind of the fact that all of these things that we're basically what this means is we're getting 4 standard deviations away from what we'd expect by just random noise. That not only do I think that viral infection that the genes we're getting it's pretty odd that we get this many genes associated with this biological function, but the fact that all of these things seem to just suggest that it's decreasing, I think makes it even more valid. And we can go into that. But I thought that was interesting. We can go to canonical pathways, and again, I can download any of these. I can download these gene lists. This is just showing you. Again, it almost looks like a lower immune system. So if a gene is green, it means it went down in the sickle cell. If it's red, it went up. You can see here a lot of this is going down. Again, I don't know if this is differences in cell type, but again, you're going to know better than I. I just want to show you one other thing so we can actually go to upstream analysis. So we've gone to biological functions overrepresented, canonical pathways, and now what we're doing is we can actually search our gene list to say, even though I don't measure MYC, this transcription regulator directly, it's interacting with 119 genes in my gene list, or it's known to. The odds of that getting all those targets of MYC by random chance is 4 times 10 to the negative 19th. Not only that, but that based on the direction of these 119 targets and what this database knows about it, it's saying that MYC is most likely inhibited in these sickle cell individuals. And basically we can go through here, and these are kind of the big players in your system. KLF1, we're showing an increase, and sure enough, it interacts with 14 targets on our list. We can take a look at that. It's known to interact with all these things on our list. The odds of that happening are very rare, number one. Number two is that things it's known to induce with the orange lines seem to be going up, red meaning up in the sickle cell. Things it's known to inhibit seem to be going down, this PCAM. I've noticed a lot of reduced cell adhesion in these hemoglobin or in these sickle cell individuals, which you might expect if the blood cells, are kind of an odd shape, which I think is fascinating. But the only one that doesn't, so this is supposed to inhibit this gene. It's going up, but that's the only one that basically counteracts the fact that this is probably more active. Given all this information, and given the fact that we wouldn't expect these targets of this particular upstream regulator by random chance, we would say that this is interacting in our system, and that the changes that is occurring with this are probably post-transcriptional and we can't measure it directly. There are some things, well actually we can. We did measure this one directly, but some of these we do not. We can actually sort this by its activation state, and we can do this here. So this would be the things that are most active in upstream regulators associated with sickle cell. The only one we measure directly is this tau1, and sure enough, we find that based on its targets, it's probably more active. Now, when things get interesting is when we go to the inhibited genes. If we look here, this ABCB6, we measured expression directly going up, but the software is telling us based on its targets, it's probably inhibited, which when I work with cancer, this is kind of what you're looking for, is that if everything worked right, you wouldn't have a disease. So you're kind of looking for these things that don't make sense. So if we actually display this as a network, it's known to inhibit all of these things, and they're all going up, which would indicate that even though it seems like the cells are trying to upregulate this, something's broke. And if this is a mutation or is this a mediator, I think this would be interesting, and I think that this is a potential target for some kind of variation or maybe modifying mutation that might affect its levels or its activity, but we are measuring an increase in its expression in these samples, although based on its targets, it's inhibited. And I want to show you one more, and that's at BNIP3L. Again, if we knock this gene out, it most resembles the gene expression you would see in a sickle cell compared to normal. And in our system, this thing is getting upregulated, 3.6. If we look at this, but again, it interacts with 11 targets. All the things it's supposed to inhibit seem to be going up. All the things it's supposed to induce seem to be going down. The only thing that doesn't follow that pattern is this one. Again, based on the gene expression evidence, I would think that this thing is inhibited, but yet the cell is trying to make more of it. So again, I think this represents a very interesting point to look for potential maybe modifying mutations that might affect the activity of this gene in certain individuals. And we can go back and I can actually look and find out what the expression was in each one of those studies. Maybe this is just a few of these individuals, I don't know. But I would think this gene and the ABCB6 or ABCB6 are potentially very interesting to look for modifying mutations. And again, we can go, there's regulatory effects. What we can also do is see basically what you're doing is you're taking upstream regulators, combining that, what targets do they interact with that also perform a particular common function. These are these regulatory networks, and these are kind of the bigger pathways that we try to build in bioinformatics. And if I click on one of these, so this will tell you the first one, but I guess what I'm trying to say is there's a lot of biological connectivity to this list, and I think this list is good. And in fact, I think we might be the first people to kind of see these types of relationships, because I don't think anybody's really done a meta-analysis with sickle cell. And again, here are the things we actually measure. Here are the upstream regulators that are probably downregulated with cystic fibrosis, and here is probably the consequences of that, which in my shape charge of blood cells, adhesions, this makes sense as far as sickle cell goes. Again, I think this is a great list. I think there's something to it. Oh, there's a PCAM one again. That I see the same things pop up, like the BMIP3L just keeps showing up in a lot of things that I look at. Let me know if you would like to pursue this farther. I can get you gene lists. I can get you spreadsheets, pathways. We can build our own pathways. I have my own bioinformatics company. I do charge for my work, but based on the market, I'm pretty affordable. So let me know if you'd like to talk about this further, and we can decide where we'd like to go. All right, thank you very much.